All right, so this lesson is going to be starting off our new unit, which is going to be genetics. Not that you care, but personally, this is one of my favorites. I just think genetics is really interesting, and I think it's kind of fun. It also has the math component when we get into Punnett squares. And so this is just kind of going to be the background. Now, you may or may not remember Punnett squares from freshman year, but normally people either really like them or they really don't like them at all. And it comes down to understanding what you're doing. So we are going to be working on Punnett squares, and we're also going to be beefing it up a little bit and going over some new genetics problems that you have not done before. And so what is genetics? Genetics is the study of heredity, which is the passing of traits from parents to offspring. And so, this is something that we see in our everyday lives. It's very relevant. You know, a lot of times from the moment a baby is born, we start looking at, whose eyes they have and whose nose they have and whose hands and fingers and toes and all of that. And it's something that's pretty interesting because we can definitely relate to it very easily. And so it's just studying how our traits pass from parents to offspring. And this was first studied by a man named Gregor Mendel. In fact, what we're going to be focusing on is what we call Mendelian genetics for part of our problems. And then you're going to have other sections that are considered non-Mendelian genetics. And so not everything is necessarily going to fit under what Mendel gave us, but pretty much the basic backbone of genetics was first discovered by Gregor Mendel. And he did that by studying pea plants. And these are just your plain old green peas that you may or may not like to eat. And so he was actually a monk. He wasn't necessarily a scientist in his day-to-day life. He was a monk. He lived in a monastery, and one of his jobs there was to tend the gardens. Now, it was through his studies with pea plants and just kind of doing some tests of his own that he discovered some of these basic genetic concepts. Now, I will tell you that Mendel got lucky because his use of pea plants, had it been other plants or other traits, it may not have worked out exactly as it did for Mendel. But luckily, he picked pea plants, and they had a lot of traits that were fitting to these Mendelian types of patterns that we're going to see. And so we do consider him the father of genetics. Now, he actually didn't get credit while he was alive. It was later, after he had passed away, that his discoveries were kind of like brought to light and expanded upon. And so in talking about the pea plant experiments, one of the things that you're going to have to understand is kind of this series of crosses that he went through. The more you understand the series of crosses and the expected results, the better off you're going to be as we go throughout genetics. And so this is where he kind of got lucky, because if you were to do an inventory of our traits, a lot of our traits are not black or white, this or that, one or the other, okay? Like if you look at hair color, skin color, eye color, height, weight, a lot of our traits, there's this whole range of what we call phenotypes. But when it came to the pea plant, all of these 7 traits that he studied were very simply inherited traits. So for flower color, they were either purple or white. Where the flower was located, they were either on the side or they were at the ends. The seeds were either yellow or green, round or wrinkled. The pods were either inflated or constricted, green or yellow. And the stems were either tall or they were dwarf. And so each one of these is going to demonstrate what we call a Mendelian inheritance pattern. And so he really just got lucky because not all traits would necessarily fit these patterns. And so he did use the peas because they were easy to work with and they were easy to cross. He had these different characteristics or traits that he could look at. Now, these pea plants did have the ability to self-pollinate. And what that means is both the male gamete-producing structures and the female gamete-producing structures are on the same plant. So even though it's sexual reproduction, the sperm of that produced on that plant can fertilize the eggs on that plant. So it's still sexual reproduction. We still have variety in the offspring because remember the gametes are going to be unique. But what he was able to do is because the plants could self-pollinate, he could kind of manipulate the crosses by just simply removing the male parts. And so something we need to understand, and we're going to see the vocabulary to this later, is that he always worked with what we call true breeding plants. Now this is what we are going to call homozygous, okay? True breeding plants could also be referred to as pure. And what that means is that if it was a purple flowered plant, it only ever produced purple flowered babies. If it was a white flowered plant, it only ever produced white flowered babies. So true breeding means it's pure, and that means that that's the only thing that that parent plant was able to produce. Okay, what we are going to call that as we go through our genetic problems is these were homozygous plants, okay, which means they have two of the same alleles, okay, either two dominant or two recessive. But essentially he started with two pure plants. Now he worked with true breeding plants of opposite character. And so what he would do is he would take a true breeding green and he would cross it with a true breeding yellow. Okay, so he would start with a homozygous green and he'd cross it with a homozygous yellow. A true breeding or pure purple was crossed with a true breeding or pure white. And he did that for each of those 7 different traits. Now what he would do so that he could kind of regulate the crosses is he would remove the male flower parts. Now on a flower, sorry my drawings are not going to be great here, but on the flower the male parts are called the anthers, and they usually kind of stick up like that, and there's usually several of them. Okay, and so we have the stamen. The whole thing is called the stamen, but it's these little anthers, these little dots at the top. This is where the pollen gets produced. And then there's usually a female part that's kind of like in the middle of those anthers. So what you can do is you can just remove these male parts, and then they can no longer self-pollinate. Okay, so he would just take away those stamen that had the pollen-producing anthers up there, and then that way the female part, he could control the pollination. So he would literally use like a paintbrush and he would kind of dust the pollen from one plant and he would place that pollen on the female part of another plant. And so he was able to kind of control the breeding, control the crosses that he was doing. So that was what allowed him to cross a true breeding green with a true breeding yellow. If he allowed them to self-pollinate, these greens would only ever produce green and the yellows would only ever produce yellows. So he kind of took control of the reproduction. So he did this for each of the 7 traits, and by crossing the plants with different traits, this is called hybridization. And so the initial generation was always referred to as the P generation or parental, okay? And so let me kind of get a pen here. So this was always my first generation, okay? And once again, in order to really kind of understand some of these Mendelian genetics patterns, we need to understand these crosses because we're going to refer back to the P's and the F1s and the F2s, okay? We're going to see them again when we get to the fruit flies and the chi-square. So the P generation was always true breeding of opposite character. It was always a true breeding of one trait. So a true breeding of trait number 1 was always crossed with a true breeding of trait number 2. Now, they were the same trait. So like purple versus white flowers, yellow versus green seeds, round versus wrinkled seeds, tall versus short plants. Okay, so that's what we're talking about there. So the P generation, or my parental generation, was always crossing 2 true breeding. So once again, we're going to learn that the word for that is homozygous. So a homozygous purple with a homozygous white. Now, the babies that are produced from that cross are called the F1 generation. It stands for first filial. And filial is like refers to offsprings or sons in Latin. So my F1 generation are going to be the babies of that parental generation. Now, if I'm crossing a purple and a white, if I'm crossing a round and a wrinkled, they're no longer true breeding. They are now what we call hybrids. Okay, so hybrid just means a cross between 2 different things. And the term that we're going to use for this is heterozygous. Okay, so all of your F1 babies are heterozygotes. Then what he would do is he would allow the F1 generation to self-pollinate. So essentially the next crossing was an F1 with itself. Okay, they're going to self-pollinate. So an F1 generation is crossed with a plant, okay? Essentially it's crossing it with itself, so that's now going to self-pollinate. And then this cross is going to give rise to my F2 babies. And this is my second filial generation. So I have my original generation, 2 true breeding of opposite character. They produce my F1s, which are all hybrids. Then I cross 2 hybrids. Essentially the self-pollination occurs, and I'm going to produce my F2 generation. Now, what's significant about this are the results, okay? Every time he did this for all 7 traits, it always came out the same. All of the F1 generation displayed 1 trait. So remember, he was crossing 2 different. He crossed a purple and a white, all the babies came out purple. He crossed a round and a wrinkled, all the babies came out round. He crossed a tall and a dwarf, all of the babies came out tall. Now, this is how we learned about dominance, okay? What we know now is a dominant trait is because of these crosses right here. Because if they were a mix of purple and white parents, why did they not come out light purple? Okay, instead they all came out one of the 2 traits. Okay, so in the F1, we get heterozygous, we get hybrids, but they all come out with one of the 2 traits. Now, the F2, when you cross 2 F1s, they always come out with the same ratio, a 3-to-1, and we need to get really comfortable with that, okay? So if you cross 2 hybrids or heterozygotes, you're going to get a 3-to-1 ratio. It's always going to be 3 of the dominant trait to 1 of the recessive trait, all right? And so as we get into the monohybrids and we start to look at our crosses, we'll see why this ratio is what it is, okay? All right, so it was from these crosses, from the P generation to the F1 generation to the F2 generation, he did this same cross with every single one of those 7 traits. Every time he got the same results, all of the F1s demonstrated 1 trait. The F2 always produced about a 3-to-1 ratio. It's from those crosses that we get Mendel's 3 laws. We have the law of dominance. So we know that that purple flower was giving a purple factor to the baby, and the white flower was giving a white factor to the baby, but yet they all came out purple. So somehow that purple one, what we call alleles, that purple allele must have overshadowed or covered up that white allele. So those babies were hybrid. They obviously had both pieces of information, but it was the purple that showed. So that's where we get the concept of dominant and recessive alleles, the law of dominance. Now the law of segregation, you have to understand that Mendel did his work before they knew about meiosis, okay? So now this is easy for us to understand. We know that we get half of our information from mom and we get half of our information from dad. And so here's the basic concept, is for every trait, you have 2 alleles. And it's because you got 1 from mom and 1 from dad. But when you go to have children, you're only going to give your child 1 of your 2 alleles, and that's because of meiosis. So essentially the law of segregation says those parental alleles, they had 2, they were separated whenever they formed their gametes. So essentially law of segregation has to do with meiosis, okay? The parent has 2 alleles, but they're only going to give their babies 1. When does that separation occur? During meiosis or gamete formation. And the last law is actually a law that we've seen before. We've seen independent assortment. And this kind of goes back to meiosis 1. Remember that when they lined up in metaphase 1, they lined up as pairs. And it didn't really matter which one was mom, which one was dad chromosome. It didn't matter which side they went on. We'd often have crossing over as well. So the crossing over could be here or the crossing over could be there. And so just the way these chromosomes lined up, the whole idea is that what a baby gets up here on this chromosome is not going to affect what the baby gets down here on this chromosome. So the inheritance of this gene is completely independent of the inheritance of that gene. Now, we are going to learn later that the law of independent assortment only applies to traits that are on different chromosomes. And this is another way that Mendel got lucky, okay? His characteristics were all on different pea plant chromosomes, and so they definitely showed independent assortment. There are times when traits could be inherited together, and that's because they're on the same chromosome. But we'll kind of talk about that at a later time. But what this is really meaning is, you think about your own traits. If you have blue eyes, do you have to have freckles? Not necessarily. You could have freckles with blue eyes, or you could have no freckles with blue eyes. So what you get for your eye color does not necessarily affect whether or not you have freckles, okay? Just because your hair is curly does not mean it has to be brown. So the genes that you get for your hair type are going to be independently inherited from the genes that you get for your hair color, okay? So that's what the whole idea of independent assortment is that if the genes are on different chromosomes, then each trait will be inherited independently from the others. So what you get on one chromosome is not going to affect what you get on a different chromosome. And so what we're going to finish out is a lot of pairs of terms. This is just a lot of vocabulary, and this vocabulary is essential to understand Punnett squares. So it was from Mendel's work. Now these were not necessarily the words that he used, but these are the words that we use today. But what we know about these terms, or how we use these terms, comes from Mendel's work. So the first two pairs of terms are genes and alleles. Okay, so a gene is a factor that determines our traits. Now keep in mind, I'm going to attempt to draw here again. Keep in mind that when we have our chromosomes, of course I'm going to draw a double chromosome. So when we have our chromosomes, a gene is just a little piece of that chromosome. Okay, it's a specific piece of DNA that's found on our chromosome. So for instance, this could be the gene for whether or not you have freckles. Okay, so the gene is just a specific piece of DNA that determines a particular trait. You have genes for freckles, you have genes for height, you have genes for eye color, you have genes for your blood type. Okay, genes determine our traits. Now, those genes can come in different forms. And so, for instance, if we're talking about freckles, the gene could say, yes, you have freckles, okay? Or that same gene could also say, no, you do not have freckles, all right? You could have another gene for eye color, and it could say blue, or it could say brown. So same gene, different versions of that gene, okay? So alleles are the different forms of that gene. This is what Mendel called factors. Now this is also why back when we covered meiosis, I told you that homologous chromosomes were not identical. The homologous chromosomes had the same genes, but where mom said yes, dad could have said no. Or where mom said blue, dad could have said brown. So those homologous chromosomes could have different alleles, which is why they are not exactly identical to each other. So genes are the different factors that control our traits. Alleles are the different forms of those genes. And it's the alleles that we represent with letters. When we get to our Punnett squares, we're going to represent our alleles with either a capital letter or a lowercase letter. And that's based on whether or not they are dominant or recessive. So a dominant allele means that it can cover up another allele. So kind of like the purple covered over the white, the round covered over the wrinkled, the tall can cover over the dwarf. And so your dominant alleles are the ones that we represent by a capital letter, and it only takes one allele. Remember, for every gene, you're getting two copies. One came from mom, one came from dad. It only takes one dominant allele for us to see that trait, okay? Now, recessive alleles, they can be covered over, and we represent these with a lowercase letter. And the only way that you can get a recessive trait is if you have two recessive alleles. Because if there's a dominant, you're not going to see the recessive allele. You can have it, but you will only see it if you have two of them. Now this right here is just an example of some common dominant traits, okay? The main thing I wanted you to see here is that dominant does not always mean more common. It's actually a dominant trait to have 6 fingers and 6 toes, polydactyly. So if you take a look at the human population, the majority of the human population are recessive for that trait. So just because it's dominant does not mean that it's necessarily more common, okay? All right, so our next pair of terms are genotype and phenotype. And once again, these are essential to understanding Punnett squares and for answering questions about Punnett squares. If you are asked for a genotype and you give a phenotype, the answer is wrong. Okay, so we have to know the difference here. So the genotype is where we use those letters for the alleles and we put them together. So what allele combinations do you have? Okay, the genotype represent your allele combinations or your genes. So you could have gotten a dominant allele from mom and a dominant allele from dad. So your genotype would be capital, capital. You could have gotten a dominant from one parent and a recessive from another parent. So your genotype could be capital, lowercase. And then you could have gotten a recessive allele from each parent, and that would be your genotype, little a, okay? So essentially your genotype are your letter combinations. So anytime it asks for genotypes, your answers or about genotypes, it's based on the letter combos. Now the phenotype is what you physically see. It's your physical trait, the physical appearance. So what color are your eyes? Are you tall? Do you have freckles? Were the flowers purple or were they white? Were the seeds round or were they wrinkled? So the actual physical trait that you can see is called the phenotype. All right, now these two words, our next pair, these describe the genotype. Okay, so I mentioned earlier we were talking about true breeding and we were talking about hybrids. These are the words that we use primarily when we're doing genetics problems. Okay, a homozygous individual, homo means the same, and this is where you have two of the same alleles. So you could have two capitals or you could have two lowercase. Now, hetero means different. So a heterozygous individual, this is what we would call hybrid, has two different alleles. So one capital, one lowercase. Now, the genotype determines the phenotype. So we can kind of put all of these words together. So we describe the genotype as either homozygous or heterozygous. And then that genotype will determine the phenotype, which is what we physically see. So if we kind of put it all together, so here we have two capital W's, that's my genotype. Because they're the same, that would be homozygous. And because they're capitals, that would be dominant. So the dominant trait in this situation was having a widow's peak. That's where your hair kind of comes down to a point on your forehead. Now here's capital W, lowercase w. Okay, so one dominant, one recessive. They are different, therefore they are heterozygous. Now remember that it only takes one capital letter to see the dominant trait. And so this person would also have a widow's peak. And then two little w's would be homozygous recessive. And it's in this case here that you actually see the recessive trait, which for this particular one means you do not have a widow's peak. So all of those are kind of like just your basic Mendelian, either you have the dominant trait or you have the recessive trait. When we talk about Mendelian genetics, what we're talking about are your single gene traits. One gene controls them, where it's a this or that situation. And this is why Mendel got lucky, because a lot of traits are not Mendelian. A lot of our traits are not this or that. Okay, a lot of our traits are controlled by multiple genes, not just one. A lot of our traits have more than just capital A or lowercase a. So if you're talking about Mendelian genetics, it's very simple. It's very easy to predict. But not all of our traits are simple dominant recessive patterns. Okay, so what we're going to look at are some other types of expression, and this is what we would consider non-Mendelian inheritance. So the first one is incomplete dominance. Okay, and when you see the word incomplete, remember that dominance covers over recessive. So in this case, it covers, but it doesn't completely cover. Okay, so in incomplete dominance, the dominant allele doesn't completely mask, and so the key to incomplete is a blend. You physically see a blending of the two traits, and this is pretty common in flowers. So one of the most common examples is a red flower crossed with the white flower gives you a pink flower. And so what happens in the case of incomplete dominance is you have a third phenotype. Okay, if it's Mendelian and it's just dominant recessive, this right here, getting an R and a W, would have been a red flower. But in the case of incomplete dominance, the red doesn't completely cover, and so we get the pink. Okay, so this is what we consider non-Mendelian because it doesn't follow that dominant recessive. So once again, in incomplete dominance, you get a third phenotype. If you crossed tall and dwarf and they were incomplete, you'd get a medium size. If you crossed black and white and it were incomplete dominance, you would get gray. Okay, so incomplete leads to a blend. Another type of non-Mendelian inheritance is codominance. And I always want you to associate co with both. Okay, so codominance is where both alleles are dominant. And if both alleles are dominant, neither one is going to be covered over by the other, so we're going to see both. All right, so this is where you would see a combination of the two traits, and you would physically see both of them in some way, whether it be spots or stripes or some other combination. So once again, flowers are often a great example. You cross a red and a white, you get a flower that has both red and white spots. Another example of codominance in us is our ABO blood type. If you get an A allele and a B allele, your blood type is AB. Okay, so that's another example. All right, multiple alleles. Now we're also going to talk about blood here because multiple alleles. Now, just to be clear, okay, don't get the word allele and gene mixed up. Okay, remember that alleles were different forms of the gene. Now, in Mendelian inheritance, you had one gene, and then you had two alleles, okay? If you're talking about Mendelian inheritance, you had the dominant allele and you had the recessive allele. And if you were to look at your combinations here, you would have 3 possible genotypes, and you would have only 2 possible phenotypes. Now, if it's multiple alleles, you have one gene, but this time there's more than 2 alleles. So in the case of blood typing, you could get the A allele, you could get the B allele, or you could get the O allele, okay? Now, as an individual, you're still only going to get 2, okay? You still get one from mom and one from dad. But what happens is, up here you had 3 genotypes, and you had 2 phenotypes. Well, we're going to really change those numbers, okay? If you're talking about blood type, let's see, AA, AO, BB, BO, OO, AB. So we now have 6 genotypes, and for this particular type of inheritance pattern, there are 4 different phenotypes. Okay, so you see all we've done is we've increased, by introducing another possible allele, we have increased variety. Okay, so now we're going to have more types of phenotypes. Okay, we're going to have a variety of physical traits that we see, more so than what you would see in a basic Mendelian type 2 allele situation. Okay, so this is just going to increase your possibilities. Now, even more than that, where you really see a wide variety of phenotypes is if it's polygenic. And if we break this word down, this literally means many. Poly means many. Genic refers to genes. So this is where you have multiple genes contributing to the trait. And so in this case, you're dealing with alleles from different genes. And so instead of just getting 1 allele from mom and 1 allele from dad, you could be getting 3 alleles from mom and 3 alleles from dad because they're all on different genes, okay? So this is where you have multiple genes controlling the trait. Now, when that happens, it creates what we call quantitative characters or a continuum or range of phenotypes, okay? So one example is eye color. Your eyes can range from light blue all the way up to very dark brown, and all the different colors in between, okay? Eye color is polygenic, and it's not always easy to predict, okay? Even though brown is dominant to blue, it's about the combinations of how many brown alleles and how many blue alleles, and that's how you end up with eye colors like hazel, or, you know, a darker blue, a lighter blue, a medium color brown, a yellowy brown, a greenish brown, and that's how you end up with all of those different eye colors, is because it's all about which alleles did you get from each parent, okay? skin tone, our skin color is like this, our height and weight. And so there's literally a range from lightest to darkest. There's a range from shortest to tallest. There's a range from lower weight to heavier weight, okay? And so what happens here is you don't just have a few phenotypes, you have a whole spectrum of phenotypes, a whole range of phenotypes. So here's an example of how polygenic inheritance works. So here's skin color. According to this, it's controlled by 3 different genes. And so you're going to get 6 different alleles. There's several combinations that you could get. So the more of the dark alleles that you have, the darker your skin tone. The more of the light alleles you have, the lighter your skin tone. And you can see that these medium color skin tones are kind of somewhere in the middle, which is why it makes sense that if you have a darker skinned person and a lighter skinned person that have a child, they're very likely to be in the middle range here. But that's not always the case. You could have a lighter skinned person, okay? See, this person would be relatively fair skinned, and you could have a darker skinned person. But all depending upon the combination, like if this lighter skinned person gave their darker alleles to their child, and then the darker skinned parent gave darker alleles to the child, you could have a child that is dark like one parent, okay? Or you could have a child that's light like one parent. Sometimes you could have 2 light-skinned parents that have a child that's darker than them. It all depends on which genes do they contribute to their child. You could have 2 darker-skinned parents who have a lighter-skinned child if they both happen to give the lighter alleles. Now, that may not be the most likely, but it is a possibility, okay? And you see instances where sometimes people can have multiple children, and some of the children have darker skin, some of the children have lighter skin, even though they came from the same parents. It's all about the combination. And so the more genes that control the trait, the more variety you're going to see in the phenotypes. And the last thing we're going to talk about is the fact that genes are not the only thing that determine our traits. Our phenotypes can also be affected by environment. Now, there are some traits that are not influenced by environment, like blood type, okay? But many of our traits are what we call multifactorial, okay? So, for instance, your personality. There may be certain things that you're genetically inclined to do, but, it could be based on your exposure, how you were raised, your weight. People's body types are kind of predetermined by genes, but how much weight you carry is also going to be dependent upon your diet and exercise. Even things like intelligence. People may have certain abilities, but if they don't have any, there's no parental help, if the parents don't encourage them to read, they don't promote schoolwork and things like that, that person may not reach their full potential. Height, improper diet. Maybe you're supposed to be, relatively tall, but if you are malnourished, you're not going to reach that potential height. And so we just call these traits multifactorial. Even things like skin tone. I mean, you can be fair skinned, but if you lay out in the sun all the time, your skin is going to appear darker. It doesn't mean you're going to pass that down to your child, but your phenotype can be affected by the environment. And that is it for our genetics. Tomorrow you're going to be starting Punnett squares, and I'm going to try to figure out the best way to kind of go about teaching you guys those. You should be getting your study guide today, so you could go ahead and start taking a look at vocabulary and things like that. But we will officially start Punnett square practice tomorrow, and we will continue over the course of the unit. So if you guys have any questions or anything, just let me know, and I hope you have a great day.